So, hello everyone. How are you? Today we'll discuss one of the important part of the CSIR-NET syllabus, that is Agrobacterium tumefaciens. Every year's question you will get sure from this topic. Agrobacterium tumefaciens is a hot topic for CSIR-NET syllabus, and you all are. In here we'll discuss all the previous year question paper of the most of the related to Agrobacterium tumefaciens. So let's start. Agrobacterium tumefaciens. So first question number one: some of the following transgenic approaches could be the use for the functional characterization of endogenous genes in plants, and the options are transformation using a binary vector containing a strong enhancer element and lacking the right border of the tDNA. You know that one of the important features of the Agrobacterium tumefaciens, or TI plasmid, that's it's have one of the important region, that is tDNA region. tDNA contain the right border and left border sequence, right border and left border, and it's a 25 base pair sequence, 25 base pair length, and there is a important another gene, another regions are there, that's a V region involved in virulency, right? Another is that here opine catabolism genes are present. So OC, or opine catabolism genes are present, right? Now here the transformation using a binary vector containing a strong enhancer element and lacking the right border of the tDNA. Now, option B, transformation using a binary vector containing a promoter-less reporter gene sequence and a selection marker gene cassette within the tDNA region. Transformation using a binary vector containing only a strong enhancer element and a selection marker gene cassette within the tDNA. Transformation using a binary vector lacking a reporter gene as well as both the left and right border of the tDNA. Now, which of the following combination can be used for transformation? We know that now we are using the ti plasmid for the transgenic plants and transgenic elements development. So ti plasmid transgenic plant development. So in case of here transformation, so it's a transgenic approach, and here ti, that is the right border and left border. You know that right border and left border is absolutely necessary for transfer, gene transfer. So transformation using a binary vector containing a strong enhancer lacking the right border. If the right border is absent, right border is absolutely necessary for transformation. For transgenic approach, it must have the right border. So option A is wrong. Option D, you can say that lacking of reporter gene as well as both left and right border of the tDNA. Left and right border is absolutely necessary for transgenic approach. So these A and D are wrong, because here right border and left border absolutely necessary for gene transfer. And right border, if any mutation occurs in the right border, that's actually it involves a lethal condition. So it will not transfer the TI or tDNA into the plant genome. So the transformation using a binary vector containing promoter-less reporter gene sequence and the selectable marker. This is selectable marker also absolutely necessary. So reporter selectable marker and reporter gene as well. This all are necessary. But right border here, as that's mentioned, the left and right border is absent or lacking. The so option D also wrong. So D wrong and A wrong. So D wrong and D wrong, A wrong. So B and C is the correct answer. Now, next question. So most of the following V proteins, there are correct functions during the agrobacterium-mediated transformation of tDNA into the plant. We know that different V regions are there, like V A, V B, V C, V D, V E, V F, and a V G region. So all have a important function. And first is that we know that what's the function of VA. VA actually acts as VA. VA acts as a receptor for acetylsyringine. VA acts as receptor, acts as receptor for acetosyringin. Acetosyringin actually a phenolic compound which is released by the wounded parts of the plants, right? That's actually acts as a chemoattractant. And so acetosyringin weed A. Now here is that weed B. Weed B actually formation of a tDNA complex or transfer complex. Weed B actually formation of the putative tDNA tDNA transfer complex transfer complex tDNA for transfer complex, right? And you can see that VC here is not there. So VD actually, or D2 actually, it's a VD actually. D have two parts, D1 and D2, and it's have a recognize the border sequence, recognize and cut the border sequence, recognize and cut the border sequence and cut the border sequence, right and left border sequence. And VD2 actually helps in induce the and a VD2 you can also see that option. So VA is not there, VB. So VB actually component of membrane structure of transfer apparatus. B1 actually form transfer apparatus. So B1 will be 2. So B1 is 2, and now C will be 2. V1 is C will be 2. Now in case of D, now D2 actually acts as an D2 VD2 recognize the tDNA border sequence, right? VD2 actually, now VD2 actually nucleus targeting of tDNA. Actually, in D2 acts as an NLS signal. D2 contain NLS or nuclear local resembles like nuclear localization signal. Nuclear localization signal, because we know that at nuclear localization, because after cutting the regions of the tDNA must have to transfer into the plant and cytoplasm and the plant and then it will enter into the plant nucleus and it will integrate into the genome of the plant. And for that reason it must have a nuclear localization signal, VD2 actually providing the nuclear localization signal, the component of membrane structure transfer apparatus for tDNA transfer. Now coming to the VF. VF actually it's a acts as a box protein. VF actually E3 ubiquitin ligase. VF as it's a act as a box protein, F-box protein. VF acts as F-box protein, or it's a E3 ubiquitin ligase. E3 ubiquitin ligase. We know that ubiquitin is absolutely necessary for proteasome mediated degradation. And we know that the two types of protein degradation machinery pathways are there. One is the lysosomal dependent pathway, another is the proteasome dependent pathway. In proteasome dependent pathway, there is ubiquitin tag or ubiquitin and tag mass absolutely necessary for protein degradation. So VF actually proteasome mediated destruction of proteins coating tDNA complex. So A will be 4. So you can VG induction of VD gene. VG actually induction of VG gene. VA and VG are actually constitutive in nature. Both are expressed. VA actually sends acts as receptor for acetylcholine, and after the VA, it is when acetylcholine bind the VA, there is a activation and cross phosphorylation occurs, and VG get activated. VG activate all other V genes, so all the V genes activated by VG. So VG actually induction of V gene. So answer is option B is the correct answer. Now next question. Now we can see that here, we can see that the all the processes. Now you can see that these are the plant cytoplasm, this is the plant cytoplasm, plant cells, and these are the agrobacterium, then these are the TI plasmid region. And agrobacterium actually, we know that bacteria have a nuclear genome as well as this plasmid genome, and plasmid actually it's a extra chromosomal self-replicating circular double-stranded DNA molecules. So it's a other than the chromosomal DNA, right? And in case of TI plasmid, there's the important part is the tDNA. These are tDNA, and tDNA contain right border and left border sequence. Now here D1 D2 come, and D1 D2 cut here, induce cut actually acts as VD2. Here we can see that VD2 and VE, VE wine also here, and the VF complex, right? VF, these are now immature T complex. Now these are pore or pillar like structure induced by VV, formation of a tDNA transfer complex, and as well as the pillar like structure or formation of conjugation tube by VV. Now here the T complex enter. Now after entry, now there is a in these are nuclear pore complex, and this is the nucleus. Now VE, VE, these are VE and VF. Now see that VE binds here, VE, and VF also going into the and now VF remove the VD and VD2. That's VD2 and VD2 and VE2. So VD2 and VE2 are removed, here is a proteasomal degradation occurs, and this after proteasomal degradation by the VF. VF actually tag the ubiquitin play important role in making ubiquitin tag, and VD2 and E2 then degraded by the proteasome, and the complex enter into the nucleus. Now here you can see that nucleus and it's also integrated into the plant genome. These are the mechanisms for tDNA transfer. Now we can see that third question, question number 3. So which are given below are 4 statements related to agrobacterium-mediated transfer of tDNA in the plant cells. Now here given below are 4 statements related to agrobacterium-mediated transfer of tDNA into the plant cells. Production of single-stranded tDNA by VD1 and VD2 protein, interaction of VE2 and VIP1 and VE3, use of VB and VD4 type 4 secretion system, activation of VA and VG complex. Now correct sequence of event from earliest to latest. So you have to arrange the sequence. So first is the activation of the VA and VG complex, right? First is the activation of VA and VG complex. These are the first one, right? So first is that, now we can see that, so if it is a damaged plant cells, these are the damaged plant cells, and these are the plasmid region. Now these are tDNA border sequence, tDNA borders, tDNA, and these are the V region. Different types of V genes are there. Now here in the this the bacterial cells, now there is a receptor receptor, it's a acts as a VA. VA actually, now these damaged plant cells, these are the damaged plant cells. Plant cells, it's release damaged plant cells, it's release acetosyringeon. Acetosyringeon then binds into the VA. Now VA, after binding here, VG also there. VG actually here some protein VG also there. VG VG become VG become autophosphorylated, and this it create after binding, this create a phosphorylation, and VG get activated, and after VG activation all the V genes are activated. So activation of VA and VG complex, these are the fast, so D will be the first one. Now see that A, you can production of single stranded tDNA by D1 and D2. D1 and D2 acts as a endonuclease. This is a CART induced CART in the right and left border. So it induced CART into the right and left border sequence. So that's actually function of feed D1 and D2. So next is you can use of VB and VD4 is type 4 secretion system. Now what do you mean by type 4 secretion system? Type 4 secretion system, it's a agrobacterium-mediated gene transfer, is an important example of type 4 secretion system. So type 4 secretion system, type 4 secretion system are type 4 secretion system, or it's a actually bacterial type 4 secretion system, also called T4SS, is secretion protein complex, secretion protein complex, protein complex found in gram negative as well as gram negative as well as gram positive bacteria, gram positive bacteria, and some archaebacteria also, it's a present. Now type 4 secretion system, type 4 secretion system, secretion system is related to, is actually or most of the cases is related or related to conjugation machinery, conjugation machinery related to conjugation machinery and DNA exchange with the actually J. Here we can see that type 4 secretion system is divided into type 4 one. So type 4 say actually as it is a using pore or pillars like or conjugation tube like structure is formed, that's actually it's also using the conjugation machinery, right, for transfer of the tDNA into the plant genome. That's why it's a type 4 secretion system. So it's a option C. And now B, integration of VE2 and VIP1 and VE3. Now integration of VE2 and VIP1. Now VIP1, what do you mean by VIP1? VIP1 is VIP1. VIP1 is VE2, VE2 interacting protein, VE2 interacting protein, VE2 interacting protein. VIP1 is known as VE2 interacting protein, VIP1. VIP1, VIP2 interacting protein 1. Now VIP1, and now VIP1 actually how it is occurs now after nuclear transfer, nuclear into when it is enters into the plant cytoplasm, and these are VD2 actually providing the nuclear localization signal VD2. Now VD2, now here you can see that the VE2 actually acts as a SSB. VE2 actually single strand binding protein. VD2 actually single strand binding protein, single strand binding protein. And now here you can VF also there. Now VF, you know that VF acts as a FVOX protein. It's a E3 ubiquitin ligase, right? VF is a E3 ubiquitin ligase. Now what's the function of VF? VF remove the VD2 and VE2, because already the DNA enters into the plant cytoplasm. Now their function is gone. VE2 and D2 function is done. Now because D2 is providing nuclear localization signal, now already it enters into the nucleus, and now they have to destroy it. So E2 and D2 are destroyed by the proteasomal degradation machinery, and ubiquitin is added in VD2 and E2 by VF. VF induce adding ubiquitin into that, and that's it's degraded. Now here some dynein protein also may be there, that's helping transfer the DNA into the nuclear pore complex. Now another important is chiropterin alpha also involved, maybe involved there, and these are actually interacting to VIP1. VIP1 is the VE2 interacting protein 1. VE2 interacting protein 1. These are nuclear pore complex. And now also there is a interaction with host cellular machinery, because agrobacterium after transfer into the or entering into the plant nucleus, they must have to interact with the plant genome. Right. There is a core histones. There is a core histones. There's a H2A1 also interacting with the H2A1. And here you can see that VF and SCF and the Qlin and ASK1, these are different proteins are there, and they induce some error prone cut at actually and into the plant genome, so that their DNA or the plasmid DNA can enters into the plant genome, and these also involve a non-homologous end joining machinery. They use the DNA repair machinery, that is non-homologous NHEJ mechanism, non-homologous end joining, after cleaving the plant genome, and then they enters the or TI plasmids or tDNA enters into the plant genome, integrated into the plant genome. These are via the non-homologous end joining. And Q80 proteins, these are several proteins that's actually involved in non-homologous end joining machinery. Q80 also involved. So there is a interactions with the host genome also for the transfer of the tDNA into the plant genome, and that's absolutely necessary. And how they are interacting, by interacting the core histone. We know that H2A, H2B, H3 and H4 are there for histones. Histone is a basic protein, and histones involved in chromatin remodeling, chromatin structural organization. So actually the it's also interacting with the core histones. It's also interacting with Qlin proteins, and it's also here Q proteins also involved. Q80 is involved, and there is a induction of double strand break in the genome of the plant genome, so that the tDNA enters into the plant genome. Now, so next we can see that here So Agrobacterium TI plasmid vector are used to generate transgenic plants. The following are example of weed gene encoded proteins that are important for the transfer of tDNA into the plants. Weed E, a single-stranded DNA binding protein. Weed E is a single-stranded DNA binding protein. Yes, weed A. Now here you have to you have to see which of the following combined protein functions inside the cell, plant cells. Now there's a we have already seen the inside the plant cells. So if you see the drawing inside the plant cell, there is a in the plant cells, and there is a the plasmid genome, and these are Ti plasmid, and the tDNA. Here the first is recognition occurs. Now tDNA cut occurs. Now after cutting, tDNA enters into the plant. Now first is the VD2 that generate T strand. Now VA that sends plant phenolic compound. These are actually outside the plant cells. So inside the plant cells is given. So these are actually not happening. So VDE is single stranded DNA binding protein. Yes, VDE VDE works in the. You can see that VDE inside the these are inside the plant, and VDE2 and inside the plant cells. VDE2 is working here. Now another is that the Weed D2 that generates T strand. Yes, this also occurring in the plant cells, and Weed F, which direct the TDNT complex protein for destruction. Yes, Weed F also working here. Weed F also working here. You can see that Weed F also in the inside the plant genome, inside the plant cells. So now here is the options. So that first is VD2. So you can B, A and D is absolutely correct. A and D is one option is there. So now VD2. VD2 actually acts as a nuclear localization signal. So VD2 also some because VD2 is a working here, also providing the nuclear localization signal. So this also inside the plant cells. Now next question we'll see. Now here the student noted the following points regarding the Agrobacterium tumefaciens. Agrobacterium tumefaciens is a gram-negative soil bacterium. Yes, it's a gram-negative soil bacterium. Opine catabolism genes are present in the tDNA region of Ti plasmid. Opine catabolism region not present in the tDNA region. It's outside the tDNA. Outside the tDNA. Opine catabolism gene are present in the outside the tDNA region. Opine gene catabolism are present in outside the tDNA region. Now, opines are synthesized by condensation of amino acid and keto acid. Opines actually maybe possible to synthesize via the condensation of amino acid. The callus structure, the crown gall. Now, first is the B is wrong. So B is wrong, and another is you can see that opines are synthesized by. Now here opine does not mention that by the plant genomes or artificially. There is no question is there. There is one question. These are not statement is not complete. Another is that a callus culture of crown gall tissue caused by Agrobacterium tumefaciens in plant can be multiplied without adding phytohormones. This also not possible. So it's a option is none of the option is correct. So it's a star mark question. Now, so now here the following are certain statement regarding the use of agrobacterium in the plant transformation. Agrobacterium tumefaciens cause crown gall disease. Agrobacterium rhizogens cause hairy root disease. Yes, it is correct. Region A and in TI plasmid responsible for replication. Region D in the A is correct. So A And in case of region D, oncogenic region of the tDNA responsible for unusual amino acid synthesis. So D also wrong, D wrong, D wrong. Now B, region A in TI plasmid responsible for replication. No, actually it's a B also wrong. So answer is A and C is the correct option 3, that the region D in TI plasmid responsible for virulency. Now next is given below are a few statements on the agrobacterium-mediated transformation of plants. Which of the following statement is correct? tDNA transfer occurs from the left border to right border. No, actually right border to left border. Right border contain NLS signal. Right border to left border, because right border have NLS or nuclear localization signal, and the GFP reporter gene can never be used for selection of transgenic plant. Yes, this is possible to use the GFP, because green fluorescent protein reporter gene can be used for selection of the transgenic plant. The transformation frequency will decrease on overexpression of the virulence gene. Transformation frequency will decrease. It will increase. Transformation frequency will increase if you overexpression of the virulence gene. So host plants genes play important role in influencing transformation frequency. Yes, host plant genes, because we have seen that the actually plants also has the agrobacterium hijacks actually plants genome. Here we can see that here there is a involvement with the host machinery, because it's also interacting with the H2A protein, H2A histone, and the VIP1, VIP1 VD2 interacting protein 1, and here also Q80 proteins also involved. So these actually also involving the host cellular machinery interacting with the host plant genome play important role in the influencing the transformation frequencies. So option D is the correct answer. Now, so next is that in the following statement were given regarding the factors influencing variation in expression level of the transgene in the transgenic plants. Now, difference in the restriction enzyme site within the tDNA region. Now here you can see that the following statement are given regarding the factor influencing the variation in expression level of the transgene in transgenic plant, difference in restriction enzyme site within the tDNA, difference in copy number of the transgene, variation in the site of the integration of the tDNA within the plant genome, presence of multiple promoters within the tDNA. So which of the following options represent a combination of statements that would not lead to variation in transgene expression level in the transgenic plants generated using the same tDNA binary vector? Now, here the same not induced. So first is that in question number 8. So first is that difference in restriction enzyme site within the tDNA. So if the restriction enzyme site, here there is no involvement of restriction enzyme. Following statement are given regarding the influence of expression level of the transgene in the transgenic plants, because so if the difference in restriction enzyme site in the within the tDNA, so there is no change in the expression level, transgene expression level. So another is that the difference in copy number of the transgene. Yes, it may be involved in expression level, because more the copy number, more mRNA, more will be the protein will be synthesized. So copy number, difference in copy number may induce in the expression level of the particular gene, and variation in the site of the integration of the tDNA within the plant genome. Yes, it's also possible variation in the site of the integration. If it is a tDNA प्लांट जीनोम में जहां भी एंटर करते हैं उसमें उसका एक्सप्रेशन लेवल में से इम्पैक्ट पड़ेगा. ठीक है, राइट. सो वेरिएशन इन द साइट ऑफ इंटीग्रेशन ऑफ द tDNA within the plant genome, presence of the multiple promoter within the tDNA, multiple promoters, because this also not because if a promoter actually it's here the promoter or a multiple promoter is not involved in the expression level. So presence of multiple promoter within the, so A and D is the option. So A and D is the correct answer. Now next question. So a set of virulence gene, a set of virulence gene, weed gene located in the agrobacterium activated by acetylsyringine. Acetylsyringine is a phenolic compound. And now it's a following some statement about the agrobacterium plant interaction. Agrobacterium tumefaciens transfer a part of the chromosome into the plant cells. Agrobacterium transfer a part of one of its plasmid DNA into the plant cells. Okay, all virulence genes of agrobacterium are inducible. All virulence genes of the Agrobacterium are functions only inside the bacterial cells. Some of the virulence genes of Agrobacterium are inducible. Some of the virulent genes functions both in bacterial and plant cells. Now, see that each of the following statement is true. Now, all the in case of Agrobacterium gene a transfer a part of its chromosome. No, actually, we know that actually in case of bacteria, this have a bacteria owns have its chromosomal DNA. Bacteria have its own chromosomal DNA, and it's also circular DNA, but the transformation occurs in the plasmid, because the TI plasmid region, that's a plasmid DNA. Plasmid plasmid DNA actually transfer occurs. So plasmid, we know that extra chromosomal. It's not a chromosomal. It's not a part of its chromosomal DNA. Plasmid actually defined as extra. Plasmid is a extra chromosomal, self-replicating, circular, double-stranded DNA molecule. So plasmid never be transferred here, right? So agrobacterium transfer a part of its chromosome into the plant cells. These are wrong. So agrobacterium transfer part of one of its plasmid DNA into the plant cells. Yes, this is correct. All virulence genes of the bacterium are inducible. No, this actually in case of some genes are polycistronic and some genes are monocistronic also there. In case of all the polycistronic genes are inducible. We know that the in here the constitutive expression constitutive expression constitutive expression occurs in weed A and weed G. Weed A and weed G actually constitutive express, and inducibles all other weed genes. Inducible is the all rest of the all other weed genes are inducible expression. Now, what do you mean by inducible? Inducible is by induction. We know that the operon and the lac operon is the inducible operon, right? Inducible, so we can induce from outside with some chemical substance, induce it. That's a inducible. So inducible operon, and like in case of, you can say that other than weed B, weed C, weed D, weed E, weed F, all are inducible genes. So all virulence genes are not inducible. So these are A and C is wrong. A and C wrong. C wrong. Now see that the B is correct. Now see, all the virulence genes of bacterial function only inside the bacterial cells. All the virulence genes function only inside the bacterial cell. No. So D also wrong. And now some of the virulence genes of the agrobacterium are inducible. Some of the virulence gene of agrobacterium is inducible. Yes, some genes are inducible. Some genes are inducible. Some genes are constitutive expression. A and VG are constitutive expression, and say constitutive expression, and VA and VG actually it's a monocystal. Weed A and weed G are monocistronic. These genes are monocistronic. We know the monocistronic actually most of the cases property of polycistronic property of eukaryotic gene, and polycistronic actually all other genes are polycistronic. Genes are polycistronic. Polycistronic actually most of the property of uh, prokaryotic cells, a prokaryotic genome, polycistronic mRNA, right? So F is the sum of the virulence genes of agrobacterium function both in bacteria and plant cells. Yes, both bacteria and some of the genes functions in both inside the bacteria and in the plant cells. So you can look at the summary, all the genes. These are actually what you have to remember for. So first of all, this one is important, and if you remember, so first is that you have to remember that all the functions of the all the wheat genes, functions of all wheat genes, how they works, what's the functions of, like wheat A, wheat A, wheat A acts as receptor for acetylserine. Wheat A acts as receptor for acetylserine. Receptor for acetylserine. Wheat B actually form wheat B actually form conjugation tube or pillar like structure. Also wheat B1 actually forming the transfer tDNA transfer complex. So conjugation tube or pillars like structure tube and transfer tDNA transfer complex. tDNA transfer complex. VC actually facilitate the activity of VD. VC facilitate the activity of VD. Activity of VD. Now VD actually acts as an, here we can write that VD actually, it's a recognize border sequence, recognize border sequence, border sequence, and it's have two function endonuclease activity and induce cartin border, and V actually VD1 and VD2. D2 contain NLS signal, nuclear localization signal. VE actually acts as SSB, acts as SSB, a single strand binding protein, and protect the tDNA to plant nucleases. Protect tDNA from plant nucleases. Plant nucleases. And VF actually it's a FVOX protein. It's a E3 ubiquitin ligase. FVOX protein. It's a E3 ubiquitin ligase. VF the E3 ubiquitin ligase. Ubiquitin ligase. And VG actually acts as transcription factor for all other virulence gene. Acts as transcription factor for all other virulence gene except VA. Except VA. So these are the important features you must have to remember. So in summary, we can say that you must have to remember what's the function of weed regions, virulence induced region, and in case of when transformation occurs, basically the tDNA region content is 25 base pair sequences must have right border and left border sequence, right border and left border. This right border and left border, among them right border is absolutely necessary. Without right border, it will never be transfer into the plant genome. Another important factor is that which portion actually occurs into the bacteria. This part actually occurs in the plasmid, and this part actually occurs in the plant genome, and these are actually pore like structures. So actually, first of all, we can see that there is a Agrobacterium tumefaciens or Agrobacterium rhizogens. They are actually free living soil bacteria. They present in the soil. When there is a wound occurs into the plants, there is some chemical release, that is acetylsyringin, and this chemical actually acts as a chemoattractant, and this chemical actually induce the receptor, that is VA. VA acts as a receptor, and it's bind into the VA, and feed A, after binding, there is a two component signaling occurs. Feed A get phosphorylated, and this autophosphorylation and another phosphorylation activate the VG. VG also get phosphorylated. The phosphorylated VG then activate all other V genes. This is actually activating all other V genes. All other V genes get activated. Now V weed B, C, D and produce. So weed B actually form the conjugation tube. These are conjugation tube so that the transfer occur, and VC facilitate the activity of VD. VD actually induce the cut here. VD actually induce cut, and VE. VE VD, VD have two parts, D1 and D2. D2 actually acts as nuclear localization signal, because the tDNA have to transfer through the pore complex or the conjugation tube into the plant cytoplasm. Now, plant cytoplasm contain many nuclease enzymes, so the tDNA may degraded by the plant nucleases, so they must have to protect the DNA from nuclease attack. So that's actually protection is done by the VE2 complex. VD2, it's a SSB. VE2, VD acts as SSB, protect the tDNA from plant nucleases. Now it enters into the, because it's contain NLS signal. Now VE2 is bind here, VD2, and now VD2, these are VD2 and E2. These are two proteins. Now they are guiding the tDNA towards the plant and a nucleus, and then the nucleus pore complex, they are present in cytoplasm. Now when they find the nuclear pore complex, then there is another protein came. The protein is VF. VF actually E3 ubiquitin ligase. VF, what's actually VF is now the D2 and E3 on, because D2 is a nuclear localization signal. So as the already tDNA enters into the nuclear pore complex, now what is the requirement of nuclear localization signal? Now nuclear localization signal should be clipped off. Another is the as the DNA entering into the nucleus, so there is no requirement of protection, because already it's entering into the nucleus. So VE2 entering into the nucleus, so VE2 should and D2 should be degraded. So VE2 and D2 is degraded by the induction, by the addition of ubiquitin tag by the VDF. VDF actually adding the ubiquitin tag into the VD2 and E2. Now VD2 and E2 protein is, all know that if the ubiquitin is added in a protein, ubiquitin, it will be degraded by the proteasome. So it will be, there is a death signal occurs, and death signal actually the E2 and D2 is degraded. Now if the D2 and E2 is degraded by the action of proteasomal degradation machinery. Now the tDNA the enters into the plant genome and integrate into the plant genome. Also, they also in tDNA also interacting with the plant cellular machinery. These are also important. Another is the plant when they enters in the plant cellular machinery. There is also there is a interacting with the plant cellular machinery complex. See that we these are actually maybe the during the transport there may be some motor protein also involved. We still do not know that which motor protein actually involved. There is a so you can see that there is a question mark in the dynein. Dynein may be involved here and intercellular transport occur. Another is that there is a one of the important protein VIP1 or VE2 interacting protein 1 is there. Another, it's also interacting with the host core H2A protein, H2A histone protein. And there is another important factor is the Q80 because they induce cut that is into the plant genome so that tDNA get integrated into the plant genome. And this induction actually by the non-homologous end joining process. So non-homologous end joining NHEJ because here Q80 protein also involved. And then these are actually the mechanisms of action, and these are very important topic for CSIR-NET syllabus. And hope we are expecting one or two questions from here. So thank you all of you.